Bacterial transformation and structure of DNA. First we discussed about bacterial transformation. Bacterial transformation. We know genes are the segment of DNA. Become evident from the work of Griffith on bacterial transformation. The scientist Griffith conduct some experiments and prove that DNA are the genetic material. Genes are the segment of DNA. The bacterium Streptococcus pneumoniae. Bacterium Streptococcus pneumoniae. With smooth colonies, Griffith used the bacterium Streptococcus pneumoniae. There are two different species of Streptococcus, smooth and rough coated. First, the Streptococcus pneumoniae smooth colonies. Smooth bacterium with the colonies injected to mice. Injected to the body of mice. The result, the mice are killed. Result, mice are killed by the activity of Streptococcus pneumoniae. Then a mutant of bacterium with a rough coating. The mutant rough Streptococcus pneumoniae is injected to the body of mice. Rough bacterial colonies are injected to mice. The mice are survived. Griffith conducted his experiment using bacterium Streptococcus pneumoniae. First, he injected smooth colonies of bacterium to the body of mice. Then the mice are killed by the disease, pneumonia. Then, next step, he injected rough bacterial colony to the body of some mice. The mice are survived. They are overcome the disease. The next step, the smooth Streptococcus colonies are mixed with rough bacterial colonies and then injected to the body of mice. Mix smooth and rough colonies of bacterium to mice. All mice are died. Mice are killed. Griffith found that smooth virulent form means disease causing species. Griffith found that smooth virulent form of Streptococcus mixed with harmless rough colonies of bacteria and inject to the body of mice. The mice are killed because by mixing with virulent smooth bacteria, the rough bacterium transformed to virulent. By mixing virulent smooth bacterium with non-virulent rough colony of bacterium the non-virulent rough bacterium transformed to virulent disease causing smooth colonies of bacterium, that is known as bacterial transformation. The result, the rough bacterium, that is non-virulent, transforms to virulent form, that is known as bacterial transformation. In 1944, MacLeod, Avery and McCarthy conduct an experiment. There is another experiment that conducted in 1944 by 3 scientists: MacLeod, Avery and McCarthy. Conduct another experiment. They prove DNA is the genetic material. DNA of virulent smooth bacteria was digested with the enzyme DNase. The first step, DNA of virulent, that means smooth, bacteria was digested with the enzyme DNase. That is an enzyme that digests DNA and injected to mice. The mice does not die. Result, the mice does not die. Once again, in 1944, MacLeod, Avery and McCarthy conducted an experiment. They need to prove that DNA is the genetic material. DNA of virulent, that is smooth bacteria, was digested with enzyme DNase and injected to the mice. The DNase are the enzymes that digest the DNA of smooth virulent disease causing bacteria. Then this mix is injected to the body of mice. The result, the mice does not die. This proves that DNA is the genetic material. Here the DNA is digested or dissolved by the presence of an enzyme that is known as DNase, and this digested enzyme mix of virulent bacteria inject to the body of mice. The mice does not die. This proves that DNA is the genetic material. DNA as genetic material. Then later Hershey and Chase, two scientists use T2 bacteriophage virus for their experiment. that act as a carrier. These T2 bacteriophage act as a carrier. Next experiment, Hershey and Chase. Conduct another experiment here. They use T2 bacteriophage as carrier. Use T2 bacteriophage virus as a carrier. Then the protein coat of virus is labeled with the protein, the all virus with a protein coat and a core material. The core material may be DNA or RNA. Here the core material of T2 bacteriophage is DNA. The protein coat of T2 bacteriophage labeled with a radioisotope of sulfur. The protein coat of T2 bacteriophage labeled with isotope of sulfur. Sulfur, that is S35. S35 is the isotope of sulfur and introduced into the bacterial cell. And introduced into bacterial cell. Radioactivity not found in bacteria. The result, radioactivity of sulfur not found in bacteria. Here, Hershey and Chase conduct an experiment using bacteriophage virus as a carrier. The protein coat of T2 bacteriophage labeled with protein coat of bacteria virus is this T2 bacteriophage is a virus. The protein coat of T2 bacteriophage labeled with isotope of sulfur and introduced into the bacterial cell. This virus is introduced to bacterial cell. The radioactivity not found in bacteria. Then in next step, when they labeled viral DNA, T2 viral DNA with radioactive phosphorus. Next step, then they labeled, the scientists labeled viral DNA of T2. This T2 virus with a protein coat and a DNA core material. First they experimented with the protein coat, adding isotope of sulfur on protein. Next step, they labeled the DNA part of this virus with phosphorus, radioactive phosphorus. Radioactive phosphorus. P32 52. P32 52. Radioactivity was found inside the bacteria. they labeled the DNA of T2 virus with phosphorus, radioactive phosphorus, then introduced to the bacterial cell. The radioactivity of phosphorus found inside the bacteria. The result, radioactivity found inside the bacteria. So they discovered DNA is the genetic material in all organisms except retrovirus. The result of this experiment is DNA is the genetic material in all organisms except retrovirus. Here we discussed detail about bacterial transformation steps and different types of experiments that prove DNA as the genetic material. Here and here the step of bacterial transformation. Next topic, structure of DNA. Structure of DNA. Here we can see the structure of DNA. It is the detailed form and this is the double helical structure of DNA. Now we learn about the detailed form of DNA molecule. Here these are hydrogen bonds, pentose sugars, phosphate molecules, pentose sugars, nitrogen bases, hydrogen bonds, phosphate molecule. The structure of DNA molecule. Each nucleotide consists of 3 subunits. Each nucleotide consists of 3 subunits. That is a pentose sugar. Each nucleotide consists of 3 subunits. They are a pentose sugar, that is deoxyribose sugar. Deoxyribose sugar in case of DNA. Deoxyribose sugar. Then the second part, nitrogen bases. Nitrogen bases. There are mainly 4 types of nitrogen bases in case of DNA. That is purines and pyrimidines. Purines. Purines are adenine, guanine. Adenine and guanine are known as purines. Adenine and guanine are purines and another pyrimidines. Pyrimidines are thymine and cytosine. The third part, a phosphate group. A phosphate group, that is PO4. These three components all together to form a nucleotide. A nucleotide part consists of three parts, that a pentose sugar, that is deoxyribose sugar, nitrogen bases. Then there are 4 types of nitrogen bases here, and a phosphate group. These three parts all together to form a nucleotide. Then what is Chargaff rule? We just denoted about one term, Chargaff rule. Chargaff's rule. What is Chargaff rule? In DNA, the amount of purines and that of pyrimidines is always equal. That is known as Chargaff rule. In DNA, the amount of purines, that is adenine, guanine, and that of pyrimidines, that means thymine and cytosine, is always equal. That is known as Chargaff rule. In DNA, the amount of purines and that of pyrimidines is always equal. That is known as Chargaff rule. A nucleoside. We discuss about nucleotide. Then next, a nucleoside. What is a nucleoside? Nucleoside. Nucleoside means base plus sugar molecule. Base, nitrogen base plus deoxyribose sugar molecule. That is known as a nitrogen base plus sugar means a nucleoside. A nucleotide means these three parts together to form a nucleotide. A nucleoside formed by nitrogen base and deoxyribose sugar. A nucleotide means base plus sugar plus phosphate. Nucleotide means nitrogen base plus sugar molecule plus phosphate molecule. We already discussed here. There are two terms, nucleoside and nucleotide. So there are 4 nucleotides in DNA. There are 4 different types of nucleotides in DNA. There are 4 types of nucleotides in DNA. That is adenine, thymine. Then thymine, adenine. Next, guanine, cytosine and cytosine, guanine. Each nitrogen base pair with phosphate and sugar. Here each nitrogen base pairs. This is one nitrogen base pair. This is another base pair. Each nitrogen base pair with phosphate and sugar. Each nitrogen base pairs with phosphates and sugars. Next, physical structure of DNA. Next part, topic, physical structure of DNA. Here, scientists Franklin and Wilkins first showed through X-ray diffraction studies of DNA that it is a double helix. Franklin and Wilkins recognized that DNA molecules with a double helical structure. Franklin and Wilkins discovered that DNA with double helical structure. DNA with double helical structure. Through the experiment by X-ray diffraction method. Through X-ray diffraction method. Franklin and Wilkins first showed through X-ray diffraction studies of DNA that it is a double helical structure. Then next, James Watson and Francis Crick were awarded Nobel Prize for working out the detailed structure of DNA. Again, James Watson and Francis Crick were awarded Nobel Prize for working out the structure of DNA according to Watson and Crick model. This is the Watson and Crick model of double helical structure of DNA. This is the detailed form. According to Watson and Crick model, DNA molecules is a double helix consisting of two strands of DNA. DNA molecule is a double helix consisting of two strands. of DNA. Next, the arrangement of the two strands is anti-parallel. The arrangement of two strands is anti-parallel. The sequence of nucleotide group up fifth to third end direction in one strand and other strand comes down third to fifth end direction. That is known as anti-parallel way of arrangement. The arrangement of two strands is anti-parallel. That is one strand goes up the direction of fifth to third end direction, and next the opposite strand that comes down from third to fifth end direction. Here we can see the anti-parallel form. This fifth to third direction and this third to fifth direction. These are two parallel strands of DNA. Next point, the backbone of helix, double helix, made of sugar and a phosphate base linked to sugar. The backbone of helix, that means two strands, helix made of deoxyribosugar, that is sugar, and phosphate molecule. Bases linked to sugar, nitrogen bases linked to sugar. Nitrogen bases linked to sugar. Next point, bases of two strands linked by hydrogen bonds. Here we can see hydrogen bonds. These are nitrogen bases. Bases, that means nitrogen bases of two strands. This one strand, this another strand, two strands linked by hydrogen bonds. Hydrogen base, two strands linked by hydrogen bonds. Adenine, that is one purine base always pair with thymine, that is a pyrimidine base. One purine base always bind with a pyrimidine base. Adenine, a purine base always pair with thymine, that is a pyrimidine base, and guanine, that is purine base with cytosine. Guanine always pair with cytosine, and adenine to thymine. that is a purine base, always pair with a pyrimidine, that is thymine. That is a pyrimidine base. The adenine, that is purine base, always pair with thymine, that is a pyrimidine base, and then the guanine bind with cytosine. Here we can see hydrogen, triple bond of hydrogen. Then 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between guanine and cytosine. Next topic, packaging of DNA. At intervals, DNA molecule is coiled around a core particle. We can see the coiling of DNA around octamer. At intervals, DNA molecule is coiled around a core particle. This core particle is known as octamer, made of 8 proteins. Each octamer coiled with DNA called nucleosome. Here we can see the structure of nucleosome. This is the octamer, that is made up of 8 protein molecule, and this octamer is coiled by DNA molecule. The DNA molecule coiled with octamer is known as nucleosome. This string is then coiled to form solenoid. This string is coiled to form solenoid. The solenoids again coiled to form supercoiled and ultimately for chromosome. Here is 4 times coiling. The solenoid again supercoiled and then ultimately to form chromosome. Here we write the points of packaging of DNA. Next, difference between DNA and RNA. In case of DNA is double-stranded molecule, but RNAs are single-stranded molecules. DNA contains deoxyribose sugar, but in case of RNA contains ribose sugar. The sugar is ribose sugar. In case of DNA, adenine complementary to thymine, but in case of RNA, adenine complementary to uracil, not thymine. Then DNA with only hereditary function. The function of DNA only hereditary in function, but in case of RNA with 2 functions. One, RNA with functions like protein synthesis. Functions like protein synthesis and hereditary material for in case of retrovirus. Retrovirus means viruses with RNA as core material instead of DNA. Such viruses known as retroviruses. Then last point, DNA duplicate its own. DNA replication take place its own. But in case of RNA synthesis on DNA template, DNA acts as a template for the formation of RNAs. Now we complete this part. If you like this class, please like, share and subscribe.